In the late 1860s, scientists believed they were on the verge of uncovering the brain's biggest secret. They already knew the brain controlled the body through electrical impulses. The question was, how did these signals travel throughout the body without changing or degrading? It seemed that perfectly transmitting these impulses would require them to travel uninterrupted by some kind of tissue. This idea, called reticular theory, imagined the nervous system as a massive web of tissue that physically connected every nerve cell in the body. Reticular theory captivated the field with its elegant simplicity. But soon, a young artist would cut through this conjecture and sketch a bold new vision of how our brains work. 60 years before the reticular theory was born, developments in microscope technology revealed cells to be the building blocks of organic tissue. This finding was just revolutionary, but early microscopes struggled to provide additional details. The technology was especially challenging for researchers studying the brain. Soft nervous tissue was delicate and difficult to work with. And even when researchers were able to get it under the microscope, the tissue was so densely packed it was impossible to see much. To improve their view, scientists began experimenting with special staining techniques designed to provide clarity through contrast. The most effective came courtesy of Camillo Golgi in 1873. First, Golgi hardened the brain tissue with potassium bichromate to prevent cells from deforming during handling. Then he dosed the tissue in silver nitrate, which visibly highlighted the nerve cells. Sorry, accumulated in nerve cells. Known as the black reaction, Golgi's methods finally allowed researchers to see the entire cell body of what would later be named the neuron. The stain even highlighted the fibrous branches that shot off from the cell in different directions. Images of these branches became hazy at the ends, making it difficult to determine exactly how they fit in the larger network. But Golgi concluded that these branches connected, forming a web of tissue comprising the entire nervous system. 14 years later, a young scientist and an aspiring artist named Santiago Ramon began to build on Golgi's work. While writing a book about microscopic imaging, he came across a picture of a cell treated with Golgi stain. Casual Ramon was just in awe of its exquisite detail, both as a scientist and an art. He soon set out to improve Golgi's stain even further and started to create more detailed references of his artwork. By staining the tissue twice in a specific time frame, Ramon found he could stain a greater number of neurons with better resolution. And what these new slides revealed would append reticular theory. The branches reaches out from each that are reaching out from each nerve were not physically connected to any other tissue. So how are these individual cells transmitting electrical signals? By studying and sketching them countless times, Ramon developed a bold new hypothesis. Instead of electrical signals traveling uninterrupted across a network of fibers, he proposed that the signals were somehow jumping from cell to cell in a linear chain of activation. The idea that electrical signals could travel this way was completely unheard of when Ramon proposed it in 1889. However, his massive collections of drawings supported his hypothesis from every angle, and in the mid 1900s, electron microscopy further supported this idea by revealing a membrane around each nerve cell, keeping it separate from its neighbors. This formed the basis of the Neuron Doctrine, which proposed the brain's tissues was made of discrete cells instead of one connected tissue. The Doctrine laid the foundation for our modern neuroscience and allowed their researchers to discover that electrical impulses are constantly converted between chemical and electrical signals as they travel from neuron to neuron. Both Golgi and Ramon received the Nobel Prize for their separate but shared discoveries, and researchers still apply their theories today. In this way, their legacies remain connected in discrete elements in a vast network of complex knowledge.